This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what are the phases of the cell cycle? The cell cycle is divided into multiple phases, including G1, S, G2, and M. Question: what controls the transitions between phases of the cell cycle? Answer: transitions between phases of the cell cycle are controlled by checkpoints, which are regulated by cyclins, cyclin-dependent kinases, and tumor suppressors. Question: how long is the shortest phase of the cell cycle? Answer: the shortest phase of the cell cycle is the M phase. Question: what happens during the M phase? Answer: during the M phase, mitosis occurs, which includes prophase, prometaphase, metaphase, anaphase, and telophase. Additionally, cytokinesis occurs, during which the cytoplasm splits in two. Question: what is the duration of G1 phase? Answer: G1 phase duration varies. Question: what is the function of cyclins? Answer: cyclins are regulatory proteins that control the progression of the cell cycle by interacting with cyclin-dependent kinases. Question: what is the function of cyclin-dependent kinases? Answer: cyclin-dependent kinases are enzymes that control progression through the cell cycle by phosphorylating specific target proteins in response to the presence of specific cyclins. Question: what is the function of tumor suppressors? Answer: tumor suppressors are proteins that act as a brake on cell division and help prevent the formation of cancerous cells. Question: what is the role of mitosis in the cell cycle? Answer: mitosis is the process by which a single cell divides into two genetically identical daughter cells. It is a part of the M phase of the cell cycle. Question: can you explain the different stages of mitosis? Answer: the stages of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible. Prometaphase is the stage where the nuclear envelope breaks down and the mitotic spindle apparatus forms. Metaphase is the stage where the chromosomes align at the center of the cell. Anaphase is the stage where the chromosomes are pulled apart and moved to opposite poles of the cell. Telophase is the final stage of mitosis, where the chromosomes decondense and a new nuclear envelope forms around each set of chromosomes. Question: what is the role of cytokinesis in the cell cycle? Answer: cytokinesis is the process by which the cytoplasm of a single cell is divided into two daughter cells. It typically occurs at the end of mitosis to complete the process of cell division. Question: what are the different types of cell division? Answer: there are two main types of cell division, mitosis and meiosis. Mitosis is the type of cell division that results in the formation of two identical daughter cells, each with the same number of chromosomes as the parent cell. Meiosis is the type of cell division that results in the formation of four genetically diverse daughter cells, each with half the number of chromosomes as the parent cell. Question: what is the role of G1 phase in the cell cycle? Answer: the G1 phase is the first phase of the cell cycle, and it is a period of cell growth and metabolic activity. During this phase, the cell grows in size, replicates its organelles, and checks for DNA damage. If the cell is damaged, it may enter a state of arrest called G1 checkpoint, otherwise it will continue to the next phase of the cell cycle. Question: what is the role of S phase in the cell cycle? Answer: the S phase, or synthesis phase, is the second phase of the cell cycle. During this phase, DNA replication occurs, and the cell's chromosomes are replicated. This phase is critical for the proper segregation of chromosomes during mitosis. Question: what is the role of G2 phase in the cell cycle? Answer: the G2 phase is the third phase of the cell cycle, and it occurs after DNA replication. During this phase, the cell undergoes final growth and metabolic activity, and it checks for DNA damage again. If the cell is damaged, it may enter a state of arrest called G2 checkpoint, otherwise it will continue to the next phase of the cell cycle. Question: what is the difference between the G1 checkpoint and G2 checkpoint? Answer: the G1 checkpoint, also known as the restriction point, is a regulatory checkpoint that occurs at the end of G1 phase. It is a point of no return for the cell. If it passes this checkpoint, it will continue to the next phase, otherwise it will enter a state of arrest. The G2 checkpoint, also known as the mitotic checkpoint, is a regulatory checkpoint that occurs at the end of G2 phase. It ensures that the cell has properly replicated its DNA and that no damage has occurred before the cell enters mitosis.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.